Welcome to a special edition of PAPERI Bright Minds, where we spotlight the pioneers shaping the future of genomics, precision medicine, and global health. As we approach PAPERI's first anniversary, we are thrilled to kick off the celebration by featuring one of the world's most influential scientists, Prof.Dr. Tishkoff.
Hello everyone, and welcome to PAPERI Bright Minds Series, where we celebrate and learn from exceptional individuals driving advancements in genetics, genomics, healthcare, and being across Africa and globally. I'm Wafa Rashid, Chair and Founder of Pan-African BGS Education and Research Initiative, PAPERI, and it's an honor to have you join us today. Today, we are thrilled to welcome Professor Dr. Tishkoff. Professor Tishkoff is a David and Lynn Slevin University Professor in Genetics and Biology at the University of Pennsylvania, holding appointments in the Perelman School of Medicine and the School of Arts and Sciences. She is also the director of the Penn Center for Global Genomics and Health Equity in the Perelman School of Medicine. Professor Tishkoff studies genomics and phenotypic variation in ethnically diverse Africans. Her research combines field work, laboratory research, and computational methods to examine African population history, the genetic basis of anthropometric, cardiovascular, metabolic, and immune-related traits, and how humans have adapted to diverse environments and diets. She plays an active role as an advocate for the inclusion of global populations in human genetics and genomics research. Professor Tishkoff is a member of the National Academy of Sciences, the National Academy of Medicine, and the American Academy of Arts and Sciences. She is a recipient of an NIH Pioneer Award, a David and Leslie Becker Career Award, a Proust Wellcome Fund Career Award, the ASHG Clark STEM Award, the Wilbur Cross Medal from Yale, the Vanderbilt Prize in Biomedical Sciences, and Penn Integrates Knowledge BIK into a chair. She is the president of the American Society of Human Genetics, is on the Scientific Advisory Board for the Packard Fellowships in Science and Engineering, and is on the editorial boards at Cell and Plus Genetics. Her research is supported by grants from the National Institute of Health, the Chan-Zuckerberg Institute, the American Diabetes Association, and the Pennsylvania Department of Health. In this session, we will explore Dr. Tishkoff's journey, her contributions to genomics, and the lessons she has learned along the way. Dr. Tishkoff, thank you very much for accepting our invitation. We were arranging this interview since February. Right? Yes, and we succeeded. We did it, yes.
Yes, and first I want to say thank you for that very, very kind and warm welcome. It is truly an honor for me to be here. I apologize. It took so long. I think that gives you a sense of what my schedule has been like lately, but I am so glad to be here.
Yes, and we are so glad. Let's start with your career trajectory. So your career spans field work in Africa through founding one of the world's largest genomic databases of African diversity. So what early experiences shaped your dedication to African genetic research?
Well, I would say it started with my interest in anthropology. So I went to school as an undergraduate student at UC Berkeley in the 1980s. And when I went there, all I knew is I wanted to be an anthropology major. And I had read books by Margaret Mead, who's a well-known social cultural anthropologist. And I said, I want to do what she's doing. And I want to do ethnographic research and things like that. So I went off to Berkeley, started out as an anthropology major. But at Berkeley, there was a researcher, a scientist named Dr. Alan Wilson. And he was the founder of what you would say is maybe molecular anthropology or using molecular tools to study human evolution. And so there were a lot of people at Berkeley who were doing that. And that got me interested in that subject. So I decided to do a double major in genetics and anthropology. And I started doing research in different labs. So one of the labs I worked in was the very first forensic lab in the country. Literally, this gives you an idea, I'm afraid, how old I am, because I worked on the very first PCR machine that was ever developed at Cetus Corporation, looking at HLA variation. I also worked with Mary Claire King, trying to identify mutations in families from Central America that had inherited deafness. So that's what got me interested initially, this interest in mixing genetics and anthropology. And then I went to Yale to do my PhD studies with Ken Kidd. And he and Luca Cavalli Sforza had a collection of cell lines from populations across the world. But they had only a few populations from Africa, actually just two. And they were two of the rainforest hunter-gatherer groups, the Mbuti and the Biaka from Central Africa. And people were using those as reference populations to represent all of Africa. It was as if those groups, any group from Africa, would just be enough to represent Africa. And for my thesis, I started studying a gene called CD4, and I was looking at variation at this gene, short tandem repeats that are highly variable and insertion deletions that are more stable, and then looking at the combination as a haplotype, how the variants are arranged along a chromosome. And that turned out to be really informative. Dr. Kidd had some collaborations. For example, he had some samples from Ethiopian Jews in the lab. He had perhaps a couple of others, but not many. And so I started reaching out to people across the globe who were studying African genetics, and there weren't many of them. And they were very kind and offered to collaborate. And when we started looking at the patterns of variation in the Africans, I noticed how much diversity there was, not just within the populations, but between the populations. They were so different in the patterns of variation compared to what we were seeing outside of Africa, where in this particular data set, people in Europe look very similar to each other. In Asia, were looking more similar to each other, but the Africans were so diverse. And that got me really, really interested in what is the type of history, the demographic history, that could result in that pattern of variation. And so when I did my postdoc, I went to Andy Clark's lab at Penn State. He was there at the time to learn more about population genetics. But before I started that, I took about a 5-month period and went to work with Trevor Jenkins at University of the Witwatersrand in Johannesburg in South Africa. And he was the one at the time who was doing really, almost the majority, I would say, of research at the time in human genetics of African populations. So it really inspired me to be in that lab and to be in that environment. And one of the things I did when I was there is I went to a meeting in Cape Town that was on the history of the Khoisan, the Click-speaking populations in Southern Africa, who traditionally have practiced a hunting and gathering lifestyle. And what was unique about that meeting is first they had people from the communities, from the Khoisan communities were there. And they had geneticists and anthropologists and historians and archaeologists, all these people from diverse fields. And I was so fascinated by these populations. And I asked people there, what would be the most interesting thing to study? And they said, you should go to Tanzania and study the Hadza and the Sandawe, who also speak with clicks in their language, but their languages are very divergent. Nobody knows how they're related to the Southern African Khoisan. And literally, that's what got me started. So as a postdoc, I applied for a grant from the National Science Foundation, together with Joanna Mountain, who was at Stanford at the time, to go study the Hadza and the Sandawe in Tanzania. And I think I had the foresight to realize, if I want to understand the history of that population, I better look at the neighboring populations because they're probably intermarrying, there's admixture. And then once I did that, I kept seeing so much variation. And then I became more and more interested in the variation. So partnered with African collaborators across a number of regions in sub-Saharan Africa, motivated partly by looking at groups with a lot of linguistic variation because that correlates with genetic diversity, to try to get a better sense of the genomic and phenotypic variation in Africa.
So your focus was on the South African region, Sub-Saharan African region, or you also studied the North Africa?
So no, I haven't up until now studied North Africa. I think it's super important, super, super interesting. And I think more should be done there. So far it's been focused more on sub-Saharan African populations, but I think it's really interesting because we're seeing that, for example, we've done work in Ethiopia, we've done some work in Cameroon studying the Fulani, and there's clear shared ancestry with people from Northern Africa. It looks like their roots, they may have originated from Northern Africa. So if we want to understand more about the history of these populations, we need to be looking at North Africa.
You know that some people classify North Africa as part of the Middle Eastern region, right?
As part of, say that one more time. As part
Middle
of the what?
Eastern region, the Middle Eastern region.
There is ancestry, right? So there's been migration and there's some shared ancestry with people from the Middle East, yes. But there's also some indigenous ancestry from Berbers, for example, who have been there for a very, very long time. So I think it's a fascinating region because of that history of, admixture and things like that.
Let me ask you another question related to your career trajectory. How did your training at UC Berkeley and Yale guide your integrative approach combining genetics, anthropology, and computational genomics?
So as I said, my training was in anthropology and genetics. So right there, I came from a very interdisciplinary background. And I think that was really important because having that anthropology foundation meant that I was studying at Berkeley. They did a traditional, they call it 4-field approach. So meaning I had to take social and cultural anthropology, biological anthropology. I took archaeology classes. I took linguistics classes. I studied primate behavior, and my honors thesis we're studying a group of baboons at the zoo. So I had this very interdisciplinary background and then was able to integrate that with genetics. And I personally think that's been the strength of my research. It's been the interdisciplinary work, and it's been partnering with people from different disciplines. I think that is always a great idea because you see things from new perspectives.
Okay. Now let's discuss about African genomic diversity and evolution. Your lab uniquely integrates genomics, proteomics, epigenetics, metabolomics, and microbiome data in diverse African environments. So what stresses and challenges come from this multi-dimensional approach?
Well, I think it's really informative because when you look at I guess you could call these things like transcriptomics, like gene expression and proteomics and metabolomics. Those are sort of intermediate phenotypes, right? So let's say that you're interested in risk for diabetes or risk for heart disease. It can be helpful to study these intermediate phenotypes, and you have a lot more power, even with smaller sample sizes, to identify genetic factors that are influencing those. But the other thing is that metabolomics and proteomics, for example, are also reflecting a person's environment. It can be reflecting their diet and other aspects of the environment. So it can really give you a clue about the role of biology and genetics and also the environment on both normal variation and risk for disease. That's why I think it's so important to integrate them, because they also influence each other. So you have to sort of look at them together. I think you're muted.
Yes. So could you share an example, perhaps from your research on high altitude adaptation in Ethiopia or lactose persistence, where environmental, culture, and genomic data illuminated human adaptation?
Sure. I actually think that lactose tolerance is the perfect example, because that there is an example where in populations that adopted cattle domestication, and that was a development of a new technology, and cattle weren't domesticated until within the past 8 to 10,000 years. This is a relatively recent thing in human evolutionary history. And so together with this domestication of cattle and then the practice of dairying, there was selection for genetic variants that enabled people to have an active form of the enzyme lactase that breaks down lactose in milk into glucose and galactose, which is taken up in the bloodstream. And so there seemed to be a fitness advantage that people who had these mutations that let them drink milk as adults, they had more children and their children had more children and so on. So these variants rapidly rose to high frequency in the populations. And some of the earliest work that we did together with our African collaborators were to identify mutations, unique mutations in East African pastoralist groups that arose independently from the most common mutations seen in Europeans that plays a role in lactose tolerance. And then when we can estimate the age of the mutation using computational approaches, we see that the European mutation is around, say, 8,000 years old, 9,000 years old. But in East Africa, they're between 3,000 to 7,000 years. And that fits in so well with the archaeological data, which shows that cattle domestication wasn't introduced south of the Saharan desert until after 5,000 years ago. So it's a great example of gene culture coevolution.
Yeah. All right. Now let's discuss an important point, health and disease insights in African populations. Let me ask you a question. Given that complex diseases like hypertension, diabetes, and obesity are rapidly increasing in Africa, so how does your work with polygenic risk scores and phenotype data help recalibrate risk assessment for African populations?
Well, that's a great question. So most of the studies of genome-wide association studies to identify genetic factors associated with risk for diseases like hypertension and diabetes have been done in people of European ancestry. Almost 90% of the individuals in, we call them GWAS, genome-wide association studies, about 90% are of European ancestry. There are a number of reasons for that. One has to do with where the research is being done, where the funding is coming from, but also because there needs to be very big sample sizes to have power when there are a lot of variants of small effect. And so that's been the justification, at least, for why people are doing this in Europeans. Excuse me. But when they calculate polygenic risk scores from these genome-wide association studies done in Europeans, and then they try to apply them to individuals who are from other genetic ancestries, they simply do not work well. They do not work, and that means that people are not benefiting from that information.
Clinical utility.
Yes, we could be giving wrong information, actually. And there could be a number of reasons. So one could be that these GWAS and Europeans, that they don't know if they have the causal mutations. They might be finding something that tags or is nearby a causal mutation. And so maybe those tagging variants are different in Africans, for example, or different African populations. So that's a possibility. There's also things like gene by gene interactions and gene by environment interactions could impact, or there could be variants that are playing a role in risk for disease that are specific to regions or populations due to demographic history and or perhaps adaptation to different environments. And if we don't study these populations, we're not going to find them. And so that's why it's so important to study more diverse populations.
Yes, sure. Your lab investigates genetic variants like APOL1 in relation to infectious diseases such as malaria and trypanosomiasis. So how is integrative genomics uncovering mechanisms of disease resistance?
So I would say in the case of APOL1 and infectious disease, I think what that is a good example of, and I would say more than, I would almost refer to evolutionary integrative genomics. So taking an evolutionary approach, because one hypothesis is that some variants that play a role in risk for disease, like APOL1, that we know is a major risk factor for kidney disease. You only see this in certain African populations, predominantly in Western Africa. Why is that? Well, it's been shown that there is a genomic footprint or a signature of natural selection, and it's known that these mutations play a role in resistance to certain types of trypanosomes that cause sleeping sickness. So the idea being that it's beneficial to have these mutations because you're less likely to get sick from infectious disease, but now it's putting you at risk for other disease. Now, this could be happening for many other diseases, right? So because genes are pleiotropic, so genes can have effects on different traits. It could be that even a gene that plays a role in resistance to infectious disease could be playing a role in risk for hypertension or for diabetes. So I think integrating an evolutionary approach is really important and helpful.
Yes. So, related to computational methods and bioinformatics, you recently examined non-coding variants' role in adaptation and disease. So, how is functional genomics guiding our understanding of regulatory elements across African populations?
Well, I think it's so important because all the existing genome-wide association studies, as well as when we do these scans of natural selection, whenever we find candidate causal mutations, I'm going to say like at least 95% of the time, they're in non-coding regions. So they're in regions that are regulating the expression of genes. Those are very tricky because it's not obvious which ones are causal. It can be very hard to identify which are in an enhancer in a regulatory region. So what we did in this recent study we published in Nature Genetics is we were interested in skin pigmentation, and we identified candidate mutations by GWAS, but also by scans of selection because there's actually a lot of variation in Africa for skin color. So if you compare populations that have very different skin pigmentation, and you ask the question, what parts of the genome are different in those populations, and which ones are near genes that are known to play a role in pigmentation, that might give you a clue about functionally important variants. Now, once you have them, we have thousands. You might get 10,000 candidate variants. Now, how are you going to narrow them down? So one way is we use something called a massively parallel reporter assay, where we look for overlap between these variants in predicted regulatory regions. And then we use a method to look at putting them into a reporter assay in a high throughput manner to look at how they're impacting gene expression. Now, once you find that, yes, they're an enhancer, yes, they're impacting gene expression, then you can use gene editing tools like CRISPR to knock out the enhancer and say, okay, if I knock it out, what's the impact on gene expression, or what's the impact on the phenotype of pigmentation? And so you can take it from starting with selection scans or GWAS and now bring it all the way to identifying causal mutations. But to do that, we typically have to do functional genomics.
Now, let me start a very important topic, ethical engagement and equity. You with great emphasis on ethical collaboration and returning results to participants. You wrote this on your website. So how has your model for community-driven consent shaped fieldwork outcomes?
That's a great question. So it's important for a number of reasons. So one is that it's the most ethical thing to do. So we of course always have to get individual informed consent, but it's always been better. We always start at the level of the community. So always, again, in partnership, we have to do this in partnership with our African collaborators because they know the culture, right? They speak the language, they know the culture. They have to be actively engaged. Okay, so together with them, going to communities, translating into the local language, and also talking in sort of layman's terms about concepts of heritability, which I think everybody does get pretty well. And then talking about the studies and why we're doing them, and what are the risks, if any, what are the benefits, if any. Talking about concerns, and only after getting community consent, then do we get individual consent and proceed. And then we try, whenever possible, to return results to participating communities because, one, it's a form of benefit sharing. So at least they're getting some information back. What did you do with my samples? And what did you learn? And, so I think just sharing that is a form of benefit, is a type of benefit. And then the other is if you want to have a long-term project and be able to return and you have to have partnerships. And we actually, one of the things we do is always include people from the community in the research itself. So as researchers, so that is important in a number of ways. One, it's training people. Two, it can be a source of income when we come and we hire people to help with that. Third, you're making sure you're doing this research in a culturally appropriate manner because you're working with people from the community. It tends to generate trust. And then following up means that if I come back and tell them, okay, this is what we've been doing, they're much more open to, there will be more welcoming in the future. And
there is a trust.
Exactly. You're establishing trust so that you can work for many, many years, not just one time go in and leave with samples, but long-term interactions and collaboration.
So let me ask you regarding if there partnership or collaboration between researcher, one in US based institutions and one African institution. So what structures or principles can ensure equitable partnerships between researcher in those different institutions, US and African one?
So I think it's important to establish the collaboration and have the discussions at the very beginning. So ideally, as the project is even being developed, so that you're having discussions about that from the very beginning and an expectation about what is going to be the role of each of the partners in this collaboration. Who is going to do what? What's going to be their role? Really, there are a couple of challenges. So one is funding. It's disproportionate, right? There's not as much that's coming to Africa. That is the truth. So we have to come up with better sources for equity in funding, right? There can be not always as good infrastructure. So sometimes there may not be certain types of equipment, for example, in some parts of Africa. And so there are a number of ways to address that. Now, in the long term, ultimately, it would be great if that was all brought there. But the reality is it's not going to be done by any single grant. That's got to be a government investment in Africa, right? So the governments have to be willing to invest in research infrastructure, right? So if they invest in research infrastructure, then there'll be that capacity there. But the other possibility is either one of the things we've done is brought people to the US for training. So coming and actually doing the research in the lab, going back with the results. So one thing is that there always has to be an MTA, material transfer agreement. So it's very clear we don't own these samples. We don't own this data. Everything gets sent back, right? And then I think one of the most important things that could be done is training in bioinformatics and computational biology, because if you have access to the internet and to computers, data analysis is the key thing. So data analysis can be done anywhere, but it is going to take a lot of training. And I think there needs to be more resources as well.
Yes, you are right. I have a question. You are now President-elect of the American Society of Human Genetics. What initiatives do you hope to champion in this regard?
I am now actually the president. So that was written before I became president. As of January, I am now the president of the American Society of Human Genetics. So way back, so this was pre-January, my goal, it's always been important to me to emphasize diversity at every aspect, diversity in the workforce, diversity in terms of training, diversity in terms of research and research subjects, right? So I wanted to make sure that was going to be my platform. That is what is truly continues to be the most important to me. Never, never, never in my wildest dreams would I imagine that what is happening now in the US would happen. My role has changed to try to preserve the efforts that we have. Most importantly, the most important job I have right now is to be an advocate for science. Because right now, science is under attack in the United States. People have lost their funding. Projects have been canceled, sometimes with no warning, because sometimes they don't like the topics of what people are studying. There should never be political interference in science. And right now, the budget, literally right now today, this week, and in the next few weeks or months, they are debating the budget for next year. And there is a proposal to cut NIH funding by about 40%. That would be devastating. Okay, it will have a negative effect for generations and generations to come. In addition, they have halted international collaboration. They have halted a lot of the research that was being done on infectious diseases, for example, in Africa. This is going to have very bad long-term consequences. So it's really important that we speak to people at Congress. I've gone to Congress. I've also written letters and tried to be very vocal about why human genetics and genomics research is so important, why NIH funding needs to be maintained at a high level, and trying to just keep the scientific enterprise going for the next generation and the generation after. I am thinking about the future generations of scientists. So that has been the most important thing that I could do right now.
Yes, this is very important in this time. Really, thank you so much for your efforts as advocate for the science, for science. And now, let me ask you a question regarding mentorship and capacity building. Your lab offers postdoc and training opportunities in computational genomics. So, what do you see as the core pillars of a sustainable pipeline for African genomic scientists?
I'm going to say one word: funding. Funding.
Yes.
Funding.
It's very important.
So I was very fortunate in my role as director of the Center for Global Genomics and Health Equity. I was reached out to Genentech. They offered a grant to be able to fund scientists with no restriction on their being a U.S. citizen, for example. So the problem is almost all of the funding has a restriction. Have to be a U.S. citizen in order to qualify. And this particular funding, no restriction, because it was from a private company. And with that, we weren't targeting just people from Africa, but based on the applicants, we ended up funding 3 people from Africa who are outstanding, one of whom is right now in my lab and working together with Dan Rader studying cardio genetics. And but that funding is ending. And now it's a challenge to find more funding, and particularly, again, in this political climate, it is extremely difficult. So I don't have an easy answer, but what we could do, even if we can't easily bring people here, we could try to put money into our grants, for one thing, so that in the grants, there could be money for funding if at all possible. So trying to bring people over here, or if necessary, us going over, and doing training locally. Or we have the internet, right? So we can do virtual training as well. But I'm not going to lie, it's a challenge. It is truly a challenge right now.
You are right. Funding is the main big challenge. Yes. So, what types of computational and system biology training would you recommend for early career African researchers?
I would just say any opportunities. I know there are a number of workshops that are offered, and I know like the Wellcome Trust does a number of workshops. And anytime that those are advertised, try to go to workshops, and sometimes they're in person, but sometimes they're virtual. There's also actually, I teach at the Jackson Labs in Bar Harbor, Maine, and they have a course on mammalian genetics. I encourage people to apply for funding because they do give out fellowships. That's a 2-week course that's held every summer in Maine. Also happens to be a beautiful location. And they'll do some hands-on. It's human genetics, a very broad human genetics training, but they do some computational training as well. So I would just say any opportunities to participate in hands-on bioinformatics, computational training. There are so many publicly available data sets right now. There are plenty of data sets that people can use as they are learning these techniques and come up with novel questions with existing data sets. So that's what I would recommend.
Yes, thanks so much. Now, let me ask a question related to the road ahead in African genomics. With over 9,000 samples in your African genomics, genomic and phenotypic diversity project, what future discoveries do you anticipate, especially around disease risk or evolutionary history?
Yeah. So again, this is not an effort done just by my lab. This has been 20-something years. Many African collaborators are key to this entire endeavor. So I'm excited also, and it's a great resource for them as well, and for everybody, I hope. But we are starting to learn more about population substructure in Africa and how populations are related to each other and times of divergence and migration events and things like that. We're integrating that data with anthropometric and cardiometabolic phenotypes, doing genome-wide association studies, and even though the sample sizes are relatively small. So nowadays, that's not a huge sample size, but there's so much variation in Africa, genetic and phenotypic, that we still get things that are informative. We look for signatures of natural selection. We find some really interesting targets, some of which are overlap with regions that may play a role in disease risk. Some of these are local to particular populations or regions. So I think there's so much to learn from this data.
How can African-led networks like PAPERI help to catalyze the application of your work into public health or precision medicine programs?
Because I just think having networks and collaboration generally is always helpful, right? Because it can introduce you to new people, new potential studies, collaborative research opportunities, funding, maybe applying for grants together, also working together with clinicians, with people doing public health, so that we can take basic science findings and then translate it ultimately into the clinic, right? So take it from the bench all the way to the clinic, and to have actual impact in the community, you have to have people who are trained in public health and who do community outreach and doctors. And I think there's a lot of potential in Africa in terms of taking advantage. Once there, I see that there's more of a movement towards using electronic health records. And as that happens, there's an opportunity to have electronic health records, perhaps merging that with genetic biobanks to move towards precision medicine, right? And that should be perhaps one of the goals.
Okay, so now we at the final reflections. What advice would you give to the next generation of African women scientists pursuing careers in genomics, evolutionary biology, or computational biology?
Well, I guess I would say don't give up. don't ever let anybody tell you that you can't do something. you can. And we're all susceptible to what they call imposter syndrome. And I certainly was, where you're just like, oh, I can never do this. Oh, I'll never be able to run my own lab. Oh, I'll never, you can, you will, you're well trained, so get that foundational training. Don't give up. Be persistent. Understand that there are going to be challenges. And I will be lying if I were saying there were no challenges. There are. It's not easy also to have work-life balance. It's a challenge every single day for those of us who have a family, have children. It's not easy. And I think having a supportive family is one of the most important things.
Yes.
That is absolutely, without that, I wouldn't be able to do what I'm doing today. That's true.
Yes, you're right. So what inspires you most today about the convergence of African genomic research, global health equity, and scientific innovation?
I'm sorry, can you repeat that one more time? I didn't hear that last part.
Okay, so what inspires you most today about the convergence of African genomics research, global health equity, and scientific innovation?
I think what's so inspiring is that there is such a wealth of information in Africa, given the most extensive genomic variation in the whole world and phenotypic variation that has just been largely unexplored. And so as we're seeing increases in technology, and more importantly, it's getting cheaper. So as the technology advances, it's also becoming more and more feasible to be doing things like whole genome sequencing, things that were impossible in the past, right? That we can now start to do. As long as we can make sure that there is good training, that the governments recognize, local governments in sub-Saharan Africa recognize the importance of this type of research and put some investment into it and build capacity, and we continue to build training efforts. There's so much that can be done. I mean, the research is not only, it's going to be important to people in Africa, right? And that way it promotes health equity, because we talked about polygenic risk scores, for example, and that, they're not effective right now in people from other ancestries. But the truth is, it helps people everywhere, the whole world. And, there are examples where you might find, because of all the diversity in Africa, a very interesting gene that plays a role in risk for heart disease or in diabetes. I think, an example, I think it's PCSK9, for example, was found because of knockout mutations that knock out the protein function that were found only in people of African ancestry. And they were associated with low LDL levels. Well, that became a target for a new drug, which is effective in all people. So it helps all of humanity. It's important for everybody. And I guess that's it. So I hope that, I'm very positive about the future of genomics research in Africa.
Yes. Professor Tishkoff, your interview will be released on July 6th, which is a very special day for us. It's PAPERI's birthday. So as we celebrated another year of championing genomics and precision medicine across Africa, would you kindly share a message to mark the occasion, a wish to PAPERI and to every initiative working to advance genomics and improve health outcomes for African populations?
Well, first of all, I want to say congratulations on your anniversary. That is fantastic. And I guess my message is keep up the excellent work. We need more people like doing what you are doing and your community is doing. It's so important. There's so many untapped resources in Africa, and the focus also on women scientists is so important. We need more women scientists, and please just keep up the excellent work that you and others are doing. That is my message.
Okay, thank you so much, Professor Sarah Tishkoff, for generously sharing your time, wisdom, groundbreaking contributions with us. Your leadership in human genetics and unwavering commitment to diversity, equity, and global collaboration have left an indelible mark on the field. It's truly been an honor to host this conversation with one of the most influential scientists of our time. Thank you so much. To our audience. Yes, okay, go on.
No, I was just going to say thank you so much. It's been a pleasure.
So our audience, thank you for joining us in this inspiring edition of the PAPERI Bright Minds Series. Today's session was more than a conversation. It was a masterclass in scientific excellence, ethical leadership, and the power of inclusive research to shape healthier future for all. So if this conversation moved you, please share it widely. Subscribe to the PAPERI YouTube channel, follow us social media, and join our growing community of change makers across Africa and beyond. Let's continue to elevate voices, build the channel, build the bridges, and advance the frontiers of science together. Thank you so much.